Welcome to the Interagency Modeling and Analysis Group Multiscale Modeling Consortium webinar series, and I'm pleased to announce that today's webinar is hosted by the Computational Neuroscience Working Group, and one of the co-leads, Dr. Ilya Rybeck, will introduce our speaker. Thank you.
Okay, I'm pleased to introduce Mark Kramer, who received his PhD in applied physics in the University of California at Berkeley in 2005. And in 2009, he received Career Award at Scientific Interface from Brooks Wellcome Fund, and get position assistant professor at the Department of Mathematics and Statistics at Boston University. So his research is focused on application of ideas and techniques from mathematics and statistics to different problems in neuroscience, and with a connection to different critical issues. And he maintains involved in many interdisciplinary collaborations, including clinical, medical, and computational research. So, today he will talk about his modeling of dynamics of seizure activity.
Great. Thank you for inviting me to participate in this webinar. It's a great group, and I'm really excited to talk to all of you about what we're up to here at BU. So I'll discuss today some multiscale dynamics of seizure activity, and this is interdisciplinary work. It involves physicists, mathematicians, and neuroscientists, statisticians at BU, computational collaborators at Brown, and clinical collaborators at MGH. So I'll start by describing the data: invasive voltage recordings from human subjects during seizure. We'll then consider how the rhythms and functional networks evolve during seizures. And in the third part of this talk, we'll think about how this activity spans spatial scales, so looking at multiple spatial scales during a seizure, and then a dynamical proposal for understanding seizure termination and a failure of seizure termination. So we'll try to cover all of these things in the next 45 minutes or so, and again, feel free to interrupt me at any point and ask questions. So let's start at the macroscopic spatial scale. We'll consider invasive EEG or ECOG recordings. So to give you a sense for that, here's a craniotomy from a human subject. The skull over the left hemisphere has been removed, and electrodes have been placed on the brain surface. And to give you a sense of scale, there's an 8x8 electrode grid in this case on the brain surface, and the spacing between the electrodes, which I'm trying to indicate with my cursor here, hopefully you can see that, is 1 centimeter. There's also, in this case, a strip of electrodes that tucks under the brain, and 2 depth electrodes that are piercing the brain and recording from deeper structures. So from all of these electrodes, voltage activity is recorded as a function of time. So in this clinical data set, we have this multivariate high-density data, about 100 electrodes on the brain surface and deep in the brain, and a pretty good sampling rate of 500 hertz. Now, the purpose of recording these data is a purely clinical purpose. It's to localize the seizure focus, or identify the area of the brain that's causing the seizure, so hopefully it can be cut out, resected, and the patient ideally cured, not suffer from epilepsy. And I'll just mention that the standard analysis of these data, again, this multivariate high-density data set, is visual inspection. So an epileptologist will stare at these time series and combine that with other imaging modalities to decide what area of the brain to remove. So again, standard analysis of these clinical data is visual inspection. And you might wonder about this resective surgery, how much brain tissue is actually removed. And I'll show you some somewhat gruesome slides. So here's a brain before resective surgery, and then here is the same brain after resective surgery. And hopefully this gives you some sense for the amount of tissue that can be removed. And one thing that our group thinks about is, can we develop quantitative tools that can improve or ideally avoid this type of surgery? So for example, can we develop techniques that can help better localize the area of resection, what area of the brain to remove, so a smaller volume of tissue could be resected? Or maybe can we think about alternatives to surgery? For example, maybe electrical stimulation of the brain or alternative drug therapies. So before we actually go on and apply some of these more quantitative techniques, let's perform our own visual inspection of the data, which is always a good place to start with a new data set. So here are some voltage recordings from a human subject, again on the brain surface during a seizure. And in this case, we're showing approximately 100 seconds of data. You can see the 10-second scale bar on the left. And in this 100-second interval, a seizure occurs. And we can ask the epileptologist, where does the seizure start? In this case, he's able to identify some region or some time interval where the seizure begins. And I've tried to sketch that here with a green line. And we can also identify seizure termination, where in this case, we see large amplitude blasts of activity as we approach termination, and then this sudden transition to a flat line. The brain essentially shuts off just at seizure termination. So what I'd like to do is zoom in on a couple intervals during the seizure and look more carefully and get a qualitative sense for what's happening in the rhythmic activity or the dynamics. So let's choose a 5-second interval just after seizure onset. And here is that interval where we're zooming in on that tiny region. And what we see is that there are rhythmic fluctuations, and we can maybe get a sense for them through visual inspection of this data. They're low amplitude, and if we compare these traces across multiple electrodes and kind of squint our eyes, we see that they look somewhat organized. If we look at these black and gray traces, we might be able to argue that it looks like organized activity is appearing across these multiple electrodes, so across space in this case. Let's move forward in time and let's choose another 5-second interval in the middle part of the seizure and investigate that. So here's a middle part of the seizure. The amplitude of the oscillations has become larger in this case. And again, if we squint our eyes and stare at these time series, we might be able to convince ourselves that the rhythmic activity has become somewhat disorganized. It's hard to say how the black and gray traces are related to each other across these multiple electrodes. Let's move towards the end of the seizure now, and again, choose a 5-second interval. I'll choose that interval here. And again, we see large amplitude blasts of activity at the end of the seizure. And you may also notice these blasts of activity are interspersed with these intervals of quiescence where not much is happening. And again, staring at this, we might be able to convince ourselves that the activity looks somewhat organized as we approach seizure termination. So the qualitative conclusion in this case, seizure activity looks somewhat organized at the beginning of the seizure, somewhat disorganized in the middle part of the seizure, and then it becomes reorganized at the end of the seizure. And we'll try to think about that more using quantitative techniques starting now. Okay. So to start, let's think about the temporal scales of a seizure and focus on the rhythmic activity. So what we're plotting here is the voltage trace from a single invasive recording, a single electrode during a seizure. And if we look at this activity, we can divide up the rhythms. And at the beginning of the seizure, the rhythmic activity tends to be fast. In this case, approximately a 20 hertz beta buzz activity. It's a low amplitude, 20 hertz rhythm at the beginning of the seizure. Now, seizure onset has also been characterized by much faster rhythms that can start at hundreds of hertz or multiple hundreds of hertz, and it may be useful for localizing seizure onset, this very fast activity, which can be much faster than 20 hertz. If we then move into the middle part of the seizure, we see that the amplitude of the activity has increased, and also the rhythmic activity has slowed. In this case, it's slowed to about 10 hertz. And if we continue this process of moving through the seizure, we can probably convince ourselves that qualitatively this activity is becoming slower and slower. We're seeing these bursts of activity become further and further apart until they reach approximately 2 hertz at the end of the seizure. So throughout the course of the seizure, the rhythmic activity tends to slow down. And this is what's called a brain chirp and is often observed in these types of seizures, which start focally and then spread to cover the entire brain. Now we can go beyond this visual inspection and compute things like time frequency spectra. So here's the time frequency spectra for a single seizure. What we're plotting here is the power, or the amplitude of the oscillations in color. Warm colors indicate high power. And if we follow the red ridge in this figure, we're showing the power, or the amplitude of the oscillation, as a function of frequency on the vertical axis and time on the horizontal axis. And again, following this red ridge, which indicates the dominant spectral activity, the dominant rhythms, we see they start around 10 to 12 hertz at the beginning of the seizure and then slow throughout the course of the seizure. Now that's just one seizure from one subject. We can repeat this analysis for multiple seizures from the same subject, and we see similar results. Rhythmic activity tends to slow during the course of a seizure. Again, this brain chirp. Rhythms tend to slow. So the seizure spans these multiple temporal scales. We can also think about seizure activity across spatial scales, and to do that, we're going to start at the macroscopic spatial scale and consider this issue of coupling. So here again is a craniotomy from an individual. There are multiple electrodes that are recording the brain's activity, and we can imagine grabbing two of those electrodes and plotting the voltage activity at those two electrodes as a function of time. So here's a 1 second interval that we're showing here, recorded at those two electrodes. And we can ask a seemingly simple question: are these two time series coupled? And if the answer is yes, we can represent that graphically by drawing a line between those two electrodes. And this analysis of coupling between brain regions during a seizure has a long history. And there are also many different options we can choose for the coupling measure. Here we choose to employ a particular measure, the cross correlation. And if you're interested in why we make that choice, we can certainly discuss that now or later in the talk. And perhaps you can imagine repeating this procedure for all electrode pairs. And in doing that, we build what's called a functional network, where the circles in this network, the nodes, are our sensors, and the edges, in this case the black lines that connect the nodes, indicate strong enough coupling between brain areas. In this case, strong enough cross correlation. So to think about how we can use these results to help us understand the seizure activity, let's start with a cartoon example of networks evolving in time. So in this cartoon, what we're showing here are a bunch of electrodes that we've organized as a column. And we've sketched potential rhythmic activity at those electrodes recorded as a function of time. And what we'll do is we'll divide up this data into smaller intervals. So we'll take perhaps a 1 second chunk of data. We'll look at the correlated activity between all of those electrodes and use that to infer a functional network. So we build this functional network for the first second of data. We then move forward in time, take another 1 second chunk of data, let's say, and infer a functional network, and so on. And we keep doing this until we cover our entire trace. So now what we have are these functional networks that are evolving in time. And to illustrate that as an example, here are some functional networks inferred from a human subject, in this case not during seizure. So this is before a seizure. And to give you a sense of orientation, we have an 8x8 electrode grid, which is indicated by this yellow square, which you can see here in our figures. So in this first figure in the top left-hand corner, we've taken the data in a 1 second interval. We've inferred a functional network, and then we're showing that functional network here, where the circles indicate electrodes. These can be surface electrodes. Actually, they're all surface electrodes in this case. Or they can be edges, and we're also showing edges, which are indicated by the black lines. So we infer this functional network for the first second of data. We then move forward in time and infer a functional network for the second of data, and then the third second of data, and fourth second of data, and so on. And we can watch as these functional networks evolve in time. And perhaps this gives you a sense that we can build these functional networks, but we're again overloaded with data. We started with this multivariate, relatively high density data set, and we've done some analysis, built these functional networks, and again, we're overloaded with information. So what we'd like to do is apply some succinct measures to characterize how these functional networks are evolving during a seizure. And I'll discuss two examples of the measures that we've looked at. So the first measure that we'll consider here is what's called the density. Hopefully, okay, here we go. So one measure we can apply to analyze these functional networks evolving in time, the density, is the number of edges in a network. And to illustrate that measure, let's consider a simple example where we have a 4x4 network I've drawn here. We could put a bunch of edges down on that network, which we indicate with lines. And in this case, we have a high density. There are lots of edges. If instead we have a network with only a lower number of edges, it has a somewhat moderate density. And if we reduce the number of edges even more, So we have that same 4x4 network. We have only a couple of edges. This network has a very low density. And again, what's nice about these network measures is they tend to have intuitive definitions. So the density is just the number of edges. We can interpret them in our particular case as a high density network. It means more correlated activity, while a low density network means less correlated activity. So let's look at this particular measure now, the density applied to a single seizure. So for reference, what we're showing here is the voltage activity recorded from a single electrode during seizure. And we can indicate here seizure onset and seizure termination for reference. Now below that, we'll plot the density. So we infer our functional networks from each 1 second interval and ask what is the density of each network, plot that density as a function of time. And that's shown below in the black curve. And what we observe when we look at this density trace is variations in the density. It appears to be high near the onset of the seizure, and maybe there are multiple peaks near seizure onset. Then the density becomes low during the middle part of the seizure, and then starts to increase again towards seizure termination. And so one interesting observation we might make is that before the seizure, if we look before the seizure, the density is about the same as during the seizure. So we have low densities before and during the seizure. Well, if we compare the voltage activity, the dynamics before the seizure, if we look at the dynamics before the seizure, we see they're low amplitude. It's hard to see any rhythmic activity here, at least at the spatial or at the voltage scale we're looking at. And compare that to the dynamics during the seizure, and we see that they're dramatically different. The seizure is characterized by much larger amplitude oscillations. So this is just one example, one seizure from one subject. We can repeat this analysis on a population of subjects, and I'll show you that result here in this somewhat busy figure. So let's try to break it down. So what we're showing here are different measures, the density and the signal energy, which I'll define in a minute, as a function of intervals. So in this case, we have 12 intervals. There's a single pre-seizure interval, 10 seizure intervals, and 1 post-seizure interval. We divide the seizures up into intervals because seizures can last different durations, even for the same patient. We want to be able to average over our population of patients and seizures. And you can think of these intervals as approximately 10-second windows of time. So if we look at the density during the seizure, which is shown as this black trace, what we find is a significant increase in the density just at seizure onset, and this is a significant increase compared to the pre-seizure level. So we see a significant increase in density at seizure onset and just before seizure termination. While during the middle part of the seizure, we don't see a significant increase in the density. Now let's compare this to the signal energy, which is essentially the total power or the total amplitude of the signal as a function of these intervals. And we find that throughout the seizure itself, the signal energy has increased. So the energy increases throughout the seizure. This is not surprising. The seizure is characterized by these large amplitude events, while the density is about the same, at least for most of the seizure, about the same as what we observed before the seizure. So the hypothesis in this case is that the seizure itself is not a uniformly hypersynchronous event. Instead, there's this coupling, this synchronization at seizure onset. There's a decoupling during the middle part of the seizure where these brain regions become less correlated, and this is consistent with animal work and observations in other human subjects. And then a recoupling just before the seizure ends. And again, this is consistent with animal work and also other observations in humans. So the coupling between brain areas seems to evolve during the seizure. As a second measure of these functional networks evolving in time, we can consider components in our network, which represent groups of electrodes connected by edges. So again, to illustrate this measure, let's consider a 4x4 network, as shown here. It's a 4x4 grid of electrodes. And we can put some edges down in that network. And we'll put them down in the upper right and lower left-hand corners. Now this defines two interesting components in our network. We have a component in the upper right corner. And we'll identify nodes that belong to the same component by giving them the same color, in this case the color yellow. And notice we can walk from one yellow node to any other yellow node by following a path of edges. So this defines one component. We also have a non-trivial component in the lower left-hand corner, and again, we'll indicate all of the nodes that belong to that component with the same color, red. And we have trivial components. These are nodes that don't touch anyone else. And notice we can walk from a red node to any other red node by following edges, but we cannot walk from a red to a yellow node. They're separate components. So in this figure and all the figures I'll show you, red will indicate the largest component. So let's apply this analysis now to an example from a single patient in seizure. So we take this patient data, we choose, in this case, a 1-second interval just after seizure onset. We infer the functional network, and then we color the components. So those components for this single 1-second network are shown in this figure in the upper left-hand corner. And what you might notice in this figure is that there's a large red component that dominates the network. It covers most of the electrode grid, the 8x8 electrode grid, as well as other strip and depth electrodes in this case. We can repeat this analysis on other 1-second networks. So here are 4 more of those 1-second networks. So again, in each case, we infer a functional network from 1 second of data, and then we color the components using the color scheme that we show in the upper right.
Yes, we heard the components. I actually have a question for you, Mark, as
Yeah.
you're putting things up. So you're saying that you inferred a network based on a second of data.
That's
So
right.
that network had correlations between those nodes.
That's right.
And then you then used that network to infer progressive signals, is that right?
That's right, yeah. So we divide up the seizure, which is maybe, let's say, 60 to 100 seconds worth of data, into 1-second chunks. And then in each 1-second chunk, we infer that functional network and then analyze its properties.
Okay. All right, and I guess you're about to tell us why you did that.
Yeah, so, right, and that can be, certainly we could spend a long time discussing that too, which I think is a really interesting area of what are the best approaches for inferring these networks and then analyzing these networks. And so one of the issues is that there are lots of different measures we can choose for coupling. We choose the cross correlation. There may be other measures depending on the system that you're analyzing that could have advantages and also their own disadvantages. And one other issue is that the way we're analyzing these networks is we're applying static measures. So we're analyzing each network individually. And really these networks are evolving dynamically in time. So developing tools that can make sense of dynamic networks in their natural state rather than splitting them up would be more effective, and that's something we're thinking about now. But for right now, I'm going to stick with the static measures. These are the common measures in the field, and really this idea of making sense of dynamic networks is an active research area that's just starting to emerge. Okay, so I introduced this notion of components, and hopefully you got to see some of that. And let's go back to the data now, where we're inferring these networks from 1-second intervals, and then looking at the components that we see. So here's a 1-second network that we've inferred, and then we're analyzing the components. And really the key point to pull out of this individual network is that it's dominated by this single red component. So all of these nodes that belong to the largest component, the red one. And as an example, here are 4 more networks that we've inferred from 1 second of data. And in each case, again, the thing to notice is that they're dominated by this large red component. So the punchline here is just after seizure onset, one component dominates. Let's move into the middle part of the seizure. And again, we can repeat the same analysis. And we find that there is a dominant component, these red components. But you might notice they've become smaller. So they're more isolated, in this case, in the lower portion of our grids. And these other components have started to emerge. So we're seeing these in different colors, like yellow and green and blue and purple. So in this case, what we're seeing is this dominant component is fractured, and these new smaller components have started to appear. If we then move to the end of the seizure and repeat the same analysis, we find that the dominant component, these red nodes, reappear. And so in this example, what this illustrates is that we're seeing this formation of a dominant component just after seizure onset. It starts to fracture in the middle part of the seizure and then reforms just before seizure termination. And again, this is just one example, one seizure from one subject. We can repeat this analysis for a population of patients and subjects, and I'll show that result here. Again, we're dividing up the seizure into essentially 12 intervals. We have 1 pre-seizure interval, 10 seizure intervals, and then 1 post-seizure interval. And what we find is a significant increase in the number of components during the middle part of the seizure. And this is consistent with the notion of the network fracturing. So our dominant component fractures, we get lots of smaller components that appear during the middle part of the seizure, and then that dominant component starts to reform and the number of components goes down. So the hypothesis in this case is that networks fracture and reform during the seizure. So the summary so far, we've looked at macro scale data. Rhythmic activity tends to slow during a seizure, so these seizures span temporal scales. And we can think about this using these pictures that we started with. So early in the seizure, we observed this fast, low amplitude activity. And we saw through a qualitative visual inspection that the activity looks organized. And in fact, we saw an increased coupling, so higher density at seizure onset, and also the formation of this dominant component early in the seizure. If we then move into the middle part of the seizure, the oscillatory activity becomes larger amplitude and it slows down. Qualitatively, that activity looks disorganized in this case, and that's consistent with the density measure. We saw the density, the coupling decrease during the middle part of the seizure, and our dominant networks start to fracture into smaller pieces. If we then go late into the late interval in the seizure, we still have these large amplitude blasts of activity. They're slower than what we had observed in the middle part of the seizure, and the activity looks more organized, at least qualitatively. And we also saw that in this density measure, this increased coupling just before seizure termination and this reformation, this coalescence of the components, again into one large dominant component just before the seizure ends. So with that, I'd like to shift spatial scales now and think about some microscale data. To do this, we're going to consider local field potential, or LFP activity, recorded from human subjects during seizure. So to give you a sense for that, what we're showing here is a cartoon representation of an electrode implantation in a human subject. And you can see here the 8x8 electrode grid, as well as this little red asterisk. And this indicates a microelectrode array. It's a 10 by 10 grid of electrodes. It's 4 millimeters by 4 millimeters on a side. It's about the size of Abraham Lincoln's head on a penny. And this tiny microelectrode array is stuck into the brain, and the clinicians and our clinical collaborators are able to record from small neural populations in the human brain in the superficial layers of cortex. So we're able to observe this local field potential activity, and we can consider this data, this LFP data, during a seizure. So I'll show you an example of that data here. So we can choose a subset of the electrodes. I think there are 9 plotted here, and we can look at the seizure activity as it evolves at this smaller spatial scale. And that's what's shown here. And if we stare at this, and again, consider a qualitative analysis, we can perhaps convince ourselves that we see a brain chirp. The rhythmic activity tends to slow during the seizure, even at this smaller spatial scale. And again, we can go beyond visual inspection and compute things like time-frequency spectra. I'm showing an example here of a time-frequency spectra where, again, we're showing frequency and hertz on the vertical axis and time on the horizontal axis. And if we look at the dominant powers, so the darkest or the warmest colors here, we see that the rhythmic activity tends to slow. So if we follow the darkest red ridge over time, it tends to slow during the seizure. There's lots of interesting higher frequency activity that I've cut off here. But if we look at the lower rhythms, which tend to dominate what we see visually, we see that they tend to slow during the seizure. And here's another seizure from that same subject. Again, qualitatively, rhythmic activity slows during the seizure. And here's another patient. Again, these LFP activity recorded from a human subject. We see the rhythmic activity tends to slow during a seizure. So even if we move down to this smaller spatial scale, rhythmic activity tends to slow during a seizure. We can also examine these dynamics across spatial scales. So to do that, I'll show an illustration of activity recorded simultaneously at 3 spatial scales during a seizure. So at the bottom of this figure is activity recorded from a single LFP microelectrode. So here we're showing the local field potential activity from approximately the middle of the seizure up to seizure termination. We can then move up a spatial scale and take the activity recorded from a single ECOG electrode and watch as that activity evolves simultaneously from mid-seizure up to seizure termination. Termination is this blue line. And we can move up to an even larger spatial scale, a non-invasive measure of brain activity, the scalp EEG. So these are electrodes directly on the scalp surface. And we can watch as this activity evolves from about mid-seizure up to seizure termination. And the thing you might notice is that across these spatial scales, something dramatic happens at seizure termination. This is perhaps most clear in the ECOG activity, this middle scale, where we have these large blasts of activity that get further and further apart, and then nothing at termination. The brain essentially flatlines. And we see something like that too in the local field potential, and also, at least in part, in the EEG activity. This is a little bit harder because there are other artifacts we have to deal with in the scalp EEG. So one question we've been considering is, can we make sense dynamically of what's happening at the end of the seizure? And in order to do that, we're going to take a brief mathematical aside here and consider this notion of critical transitions, again, with the goal of understanding what's happening dynamically at seizure termination. So what is a critical transition? We can think of this as a tipping point in which a system shifts abruptly from one state to another. And to illustrate that idea, here's a cartoon example. So imagine we have a potential well that I've drawn here, and we can drop marbles in this potential well. And I'll start with that red marble, which hopefully you see in the middle of your screen. And if we let that marble go, it rolls around and eventually lands in this trough and settles down. So this is a fixed point. If we start the marble there, it stays there. We can give that fixed point a label. I'll call it A. And if we imagine kicking this marble to the left or right, it rolls back. So this is a stable fixed point. If we give it kicks to the left or right, it returns. Now we can drop marbles in other places in this potential well. So I'll start one at the far right of this potential well. We let it roll around and eventually it comes to rest at another fixed point. I'll give this a label B, and it's also stable. If we kick this marble to the left or right, it comes back to this fixed point. There's a third fixed point in this system. It's at one of these relative maxima in our potential well. If we start the marble at that green position, it stays there. It won't roll away. But if we give it a kick to the left or right, it does roll away. So this is an unstable fixed point. And a simple way we can summarize this dynamical system is just list the number of fixed points. In this case, there are 3. And now let's imagine we have a parameter that we can tune, and we change this parameter. And as we do that, we deform the potential well. So we start changing the conditions in our system. We're changing a parameter, and our potential well deforms. And we can ask a qualitative summary of this system, how many fixed points are there? So we change the conditions, the potential well deforms, and we see in this case there are still 3 fixed points. We have our stable fixed points A and B and our unstable fixed point in green. So we still have 3 fixed points, even though qualitatively the potential well has changed. Let's keep doing this. We keep turning our parameter, and we see that the potential well deforms. Here in this case, we still have 3 fixed points, 2 stable and 1 unstable. So qualitatively, we can list the number of fixed points at the left in our figure. We still have 3 fixed points in this case. And let's change this parameter just a tiny bit more, and what we find in this case is a dramatic change in our system. Instead of 3 fixed points, there's only 1. So what's happened here is we've qualitatively changed the dynamics in our system. We've undergone a bifurcation where we've gone from 3 fixed points to 1. We've crossed a tipping point in this system. And our proposal is that we can think of seizure termination in a similar way, as this critical transition, this sudden change in the attracting state of the brain. So to make this a bit more concrete, let's consider briefly an example motivated by neuroscience, the Wilson-Cowan equations. And maybe this system of equations is familiar to some of you. It's a mean field model of neural population activity. It consists of 2 populations, an excitatory population and an inhibitory population of cells. The excitatory population activates the inhibitory population, while the inhibitory population inhibits the excitatory population. And we can write down this model as a system of differential equations as shown here. We have these E and I variables that represent the 2 populations. And this S function is a sigmoid, which is shown here at the right. Now, we'll use this model to illustrate some of these notions of critical transitions. And what's nice about this model is that we can visualize it. It lives in a 2-dimensional space, so we can look at it. It's easy to look at. And it's also applicable. It's arguably related to the biophysics of the brain. And we're going to start by considering this model as we just a single parameter P, which controls the excitability of our excitatory population. So let's look at this model, and let's start by drawing some pictures. So we'll fix this parameter P, and we'll draw the phase space of this system. So the phase space, we can represent the system by looking at the 2 variables. Here we're plotting the E variable versus the I variable, so the dynamics of our 2 populations. And what we find when we look in this phase space is that there are 2 attracting fixed points. These are indicated by the black circles. We can see that these are attracting fixed points by looking at the direction field. So if we look at the arrows drawn in this picture, and we start somewhere else in this two-dimensional plane, we get pushed to one of these fixed points just by following the arrows. There's also a repelling fixed point, and that's at the center of our figure. And this repelling fixed point has a stable manifold, this red line. And this red line acts as a separatrix. If we start to the left of this red line, we go to our lower fixed point. Well, if we start to the right of this red line, we end up at the higher fixed point. And these fixed points have a biological interpretation. The attracting fixed points have a biological interpretation. We can think of one of these attracting fixed points as a rest state. Both E and I are near 0. And the other fixed point as an activated state, where both populations, the excitatory and inhibitory populations, have non-zero values. So let's consider now increasing that parameter P. Let's increase the drive to our excitatory population and see what happens. So here we've increased that excitatory drive a little bit, and there's a subtle change that's happened here. Our repelling fixed point has moved a little bit to the left. And the separatrix, the whole system, the separatrix has moved a little bit to the left. If we can continue increasing this drive to the excitatory population, we see that that repelling fixed point moves a little bit more to the left. And if we keep doing this, we eventually see a dramatic change in our system. Instead of having 3 fixed points, these 3 black dots, we have only 1. So only 1 fixed point remains. And so somewhere between these parameter values, between this drive of 0.2 and 0.3 to our excitatory population, a bifurcation occurs, a dramatic change, a tipping point in our system. So we can think more carefully about that bifurcation, and we can look at things like the eigenvalues in our system. And if we look at the eigenvalue of one of our rest states, we see that one of the eigenvalues goes to 0, which is exactly what we expect as we approach this bifurcation. And we can compute things like bifurcation diagrams for this system. So what this bifurcation diagram is showing is the attracting states of one of our variables, the E variable, as a function of our parameter. And these bifurcation diagrams are useful because we can identify exactly where this sudden transition occurs. In this case, we can identify the value of our parameter P where this transition occurs. And it's somewhere between 0.2 and 0.3. In this case, it's at 0.28. And we can think about how changing this parameter affects this attracting state in our system. So if we're very far from this bifurcation, so we start far from this bifurcation at a small value of P, and then we change P by a little bit, we only change our attracting state by a tiny bit. If instead we're close to the bifurcation, and we change P by a small amount, the drive to our excitatory system, we get a dramatic change in the attracting state. We fall off of this lower branch of attracting states and swing up to this upper branch. So a small change in P causes a dramatic change in our system. And that's the punchline here. So near the bifurcation, small changes in our parameter can give rise to enormous state changes in our system. So we can also add noise to this model and consider the activity of the excitatory state as we approach this bifurcation. So let's start very far from the bifurcation. Again, we're simulating the system with a certain amount of noise. And what we find is that the system, this E variable, shown here in blue, fluctuates around the resting state, which is shown in red. And we can get a sense for the extent of these fluctuations. Let's now change the parameter in our system and move closer to the bifurcation. We give the system the exact same noise, and what we find in this case is the system still fluctuates around the rest state, but that these fluctuations become larger. And what this observation motivates is that as we approach this bifurcation, but before we hit it, something in our system changes. In this case, we see that the extent of the fluctuations has increased. And hopefully what this motivates is that there are dynamical warning signs of this impending transition. So in this lower figure, we're near the bifurcation, but we haven't crossed it. And so we can look for some of these warning signs, these changes in the dynamics of our system before we hit the bifurcation. And I'll just briefly describe some of those warning signs here. So some things we can look for critical slowing down in our system. That means near the transition, the system becomes increasingly slow in recovering from perturbations. And that makes sense mathematically because one of the eigenvalues is getting closer to 0. And this manifests in a measure we can apply to data. We can look at things like the autocorrelation. And if we're far from the bifurcation, we can compute the autocorrelation of our system, so we can look at the dynamics of the variable E. And here we're showing the autocorrelation as a function of lags. We can choose a particular lag value. I've chosen here arbitrarily a value of 25 milliseconds and look at the autocorrelation. If we then adjust our system and get closer to that bifurcation and evaluate the autocorrelation at a particular lag, we see it increases. We can get even closer to the bifurcation, so we keep increasing the drive to our system, and we look at the autocorrelation at a particular lag and we see it increases. So the punchline here, as we approach the bifurcation, but before we cross it, we expect this particular measure that we can apply to data, the autocorrelation and non-zero lag, to increase. Another measure we can look at that manifests this critical slowing down is the power spectrum. And we can repeat the same analysis. Let's simulate these Wilson-Cowan equations, compute the power spectrum, and then look at the power at low frequencies. If we're far from the bifurcation, we have a particular power value at low frequencies. If we move closer to the bifurcation and even closer, we find that the power at low frequencies increases. This is perhaps not a surprise. The autocorrelation and power spectrum are Fourier transform pairs. But as we approach the bifurcation, the power at low frequency starts to increase. So again, before we hit the bifurcation, we can expect to see some of these features. Another warning sign we can look for is flickering. And this manifests when the system moves back and forth between the two alternative attractors before shifting all the way to 1. And to illustrate this idea, let's consider again the phase space for our Wilson-Cowan equations. And let's imagine we start the system at the lower fixed point, this rest state. And we give our system some noise, so this rest state experiences some kicks. And it gets pushed a little bit to the left and right in the system, and eventually it gets a large enough kick that can send it across the separatrix and to the alternative attractor. And now the system will also have noise in this state. It'll get kicked around and eventually can get kicked back across the separatrix back to the lower state. And as a simple way of representing the dynamics of this system, we can plot the position as a function of time. And so the position will just represent is the system in the rest state or the active state as a function of time. It starts in the rest state, it gets small stochastic kicks. Every once in a while it gets a large enough kick, goes across the separatrix and sits in the active state. And it stays in that active attractor for a certain amount of time until it gets a large enough kick to go back across the separatrix to the other rest state. And in this way, the system flickers back and forth between these two alternative attractors as a function of time. So let's return to our ECOG data and ask whether or not we can see any of these dynamical signatures of an impending critical transition in the data. And we can start with visual inspection. So here's a single voltage trace from one of these macroscopic electrodes as a function of time. We can ask questions like, do we see features of critical slowing down? Well, we've discussed this notion of the brain chirp, the slowing rhythmic activity, so that's perhaps consistent with this notion of critical slowing down. What about the autocorrelation? Do we see increased autocorrelation? That's hard to say just by looking at these data, and we need to actually apply these measures to the data and check that. What about flickering? Do we see a manifestation of two alternative attractors as we approach seizure termination? Well, maybe. And let me try to suggest that here. So we can characterize the seizure state in a very crude way. We can consider it as a high variance state. And I've tried to indicate that high variance state with this vertical red box, which has a large vertical extent. Now let's compare that to the post-seizure attracting state, which we've indicated here. The brain activity is essentially flatlined. It has a very low variance, and we're using here this thin gray or green rectangle to represent that post-seizure state. And if we look just before seizure termination, we do see some manifestations of both attracting states. We see these large amplitude blasts of activity that are consistent with the seizure state, and then these low amplitude intervals that are consistent with the post-seizure state. So we're seeing manifestations of both attractors, at least qualitatively, in this picture. We can go beyond this visual inspection and consider quantitative analysis. I won't go into that here, the details, but I'll just mention that we do see signatures of a critical transition in the approach to seizure termination. And we see them across the spatial scales, in the scalp EEG, in this macroscopic ECOG data, and in the more microscopic LFP data. And if you're interested, I'd be happy to, the reference is here, and I'd be happy to send along more information. And maybe, see we have maybe just a few more minutes. I'll just quickly go through the last part, and I'll just summarize what's here, and if you're interested, we can talk about it in more detail. So here we have some evidence of a critical transition. We can use the data from a population of patients and seizures to suggest that there's a critical transition at seizure termination, but we can't prove it just by looking at data. What we can do is develop a mathematical model to try to simulate seizure termination. Here I've put simple in quotes. We'll consider a simple biophysical model of seizure activity, and we'll consider a mean field model. And the reason we do that is to try to match the spatial scale of the data we observe. We don't really know what's happening at the single neuron level in these LFP recordings. We don't observe individual units in this case. We're not patching a cell and seeing what's happening. So we're instead modeling the data that we have in hand, the field activity. We can analyze these model dynamics. I'll just quickly go through this and we can come back and talk about it later. We can induce a seizure attractor, which I've represented here as a ring. It is a high variance state. We can change the conditions in our system and induce a post-seizure attractor, which is a point in this two-dimensional space. And this is consistent with a low variance state, the post-seizure state. And what's nice about this model is we can induce bistability, so we can see both attracting states in our data. And we can induce things like flickering in our system, moving back and forth between these two alternative attractors. This is a cartoon sketch that I've drawn here. If you're interested in the mathematical details, we can compute things like bifurcation diagrams. I won't go into the details here, but what's nice about these bifurcation diagrams and modeling in general is we can identify exactly the nature of the critical transition. In this case, it has a particular form, which we can discuss if you're interested. And I'll just mention that this model is able to mimic all of the features we see in the clinical data. A reasonable question is, so what? Why bother building these models in the first place? Well, hopefully what we can eventually do is relate these dynamical mechanisms to biophysical mechanisms. So here's an outline of potentially that approach. We've thought carefully about, well, maybe I'll start by mentioning that there are many proposed biophysical mechanisms of seizure termination, whether it's changing ion concentration, synaptic transmission, or reduced pH. Some of those are listed at the bottom of the figure. And what we've focused on is a dynamical principle of seizure termination that's consistent with these multi-scale data. So perhaps we can use this dynamical principle to constrain the underlying biophysical mechanisms. And this requires modeling if we want to try to connect these biophysical mechanisms back up to dynamical principles. Also, we might think about treating the seizure as treating the dynamical mechanism and using whatever means are convenient or useful for a particular patient to treat that dynamics. So using whatever biophysical means we can to treat the dynamics of a seizure. And maybe I'll stop here and just mention that we can use the model to make a prediction. Status epilepticus is when a seizure fails to terminate. In our model, the self-terminating seizure goes through a particular critical transition. And what we've proposed is that a status epilepticus, when the seizure doesn't stop, it's when we fail to cross that transition. That should manifest as certain dynamical signatures, and I'll just go to the last slide here and mention that we see all of those signatures. So our model is able to make this prediction about what we should see in status when a seizure fails to stop, and that is consistent with what we see in the data. So to conclude, we've thought about seizure activity across spatial and temporal scales. Rhythmic activity tends to slow. Brain areas tend to fracture and coalesce during the seizure in a very stereotyped way. And perhaps we can think about the dynamical mechanisms of seizure termination. And I'd like to thank all the patient volunteers as well as our funding sources and the collaborators and all of you for participating in this webinar.
Thank you very much, Mark. This is very interesting. We're now open to have any questions. So I guess, Mark, I'll start.
Sure.
I found your component analysis really interesting, and I was just wondering if you used it on non-seizure data and if there are networks that actually change when there's no seizures, or that 1 second segment, is it really stable when there's no seizures?
Yeah, so we've looked at that in a couple of different ways. So we've considered long chunks of non-seizure activity, and we tend to use we make the choice of 1 second to balance two things. We want enough data to make accurate inference about the coupling, but we don't want so much data that the coupling could change over the window. So we see 1 second as kind of a compromise in this case. And if we build those 1-second networks, and instead of doing it for a seizure, let's do it for 24 hours. So let's build these networks over time. What we've seen in invasive ECOG data and in scalp EEG is that those the networks tend to be remarkably stable. We tend to see similar networks, especially when we average over 100 seconds of data or maybe 2 minutes of data. Those network features tend to be very consistent no matter when we look. And that's something we don't understand why that's the case. Maybe it's related to the anatomy and that's not changing so much over a 24-hour interval. Maybe it's related to something else. But those, the maybe most global features of the networks tend to be stable when you average them over long enough times. Moment to moment, there's variability, but when averaged, they're remarkably stable.
So I wonder if, I don't know, in the future, if you can impose those networked correlations during the seizure, you create stability.
Yeah, it's a really interesting idea. If you have that ECOG grid implanted and you can record from it and send voltage activity or some drug delivery through those individual nodes, what you could do either to try to stop the seizure from occurring or once you see it start to maybe end it right away. It may be hard to stop the seizure from occurring because it's hard to predict when a seizure will start. And so it may be the case that you have to detect that it started and then try to stop it as quickly as you can rather than prevent it from starting. But yeah, I think those types of ideas are really interesting.
So there is no anything that could predict in advance when it starts?
It's so my sense is that it's a very difficult problem, and lots of people have been working on it. I think there was a company recently that had an implanted device. I forget what the company name is. I think they were in Australia that showed they were able to get pretty good prediction, but it's a difficult problem.
So based on what? Also, if they based on EEG or what?
Yeah, so that's, people have tried it usually with those types of measures, so using something like EEG or an implanted electrode, so maybe directly on the brain surface. There are other ways to predict seizures. Patients have auras, so they may have a sense of an impending seizure. There are seizure dogs that can detect when a seizure is going to occur, probably through some other physiological change that's going on in the individual.
So Mark, in the beginning you showed us the gruesome brain pictures, and you're saying that your hope is that your work would try to reduce the amount of brain that's removed.
Right.
So would you be able to maybe localize seizures better?
Yeah, so that's one of our hopes, is that we can use these techniques to identify the features of the network that can be exploited to try to either stop the seizure from occurring or maybe quickly end it when it starts. And so we're trying to do that in a couple of ways. We're trying to figure out where in the network this thing starts and then spreads. And maybe you can cut out the part where it starts, or maybe you could try to firewall that part if you can't cut it out. Maybe it's part of eloquent cortex, so it's responsible for speech or movement, and you can't resect it. But you could try to firewall it in some way and prevent it from spreading in the network. And part of that is this prospective studies where we kind of predict where that's going to happen and then follow up with patients over time to see after the resection whether or not they become seizure-free, and that's independent of our work. That's determined by the physicians what's cut out. And also developing methods to track communities dynamically in time. So if we can imagine this seizure is starting somewhere and then spreading, we'd want to kind of try to track it backwards in time and see where it starts. And so we're trying to develop techniques now to track these communities, these evolving communities in time through these time index networks. Oh, I think Raj is saying something, but I can't hear anything.
Can you hear me now?
Yeah.
My mic was off. Okay. So one of the things that I saw at the Network Brain Symposia before SFN this year was a lot about resting state networks and fMRI and DTI data of resting state networks. And one of the things that struck me there was that in the resting state networks, that when they're looking at the whole brain, right? I mean, voxelated and averaged and so on. What they find are there are certainly communities of activity, localized activities, but there are also fairly long-range connectivities, so to speak, across many regions. And in terms of delinking them, you wouldn't really go after the ones that are highly connected, but you would go after the one node that has only 2 connections, but that's the only node that connects 2 communities. So from a thinking of it from a network point of view, what is communication across a network is sensitive to is not necessarily sensitive to the one that talks to a lot of things or where things started, but rather the linking things. And some of data was showing that those linking nodes could be places like where multiple regions sort of connect. So they were talking about the 4-corner, 4-state junction kind of a models, multiple areas. They're all very localized, but then they all go through this very narrow area to talk to each other or talk to other things. So it seems like there are opportunities there to connect to what you're talking about with those resting state networks, and then say, not really look at where did things start, but rather which very localized, small, narrow areas that you can take out to disrupt the spreading or connectivity or things like that. Or recurrence, right? Disrupt the recurrence, for example. That is a big issue.
Yeah, I think that's a great idea. And there are particular network measures you can apply to assess just that. So the betweenness measures how between a node is between other things.
Right.
So exactly as you described, if you think of like a dumbbell, you have lots of connections on one end of the dumbbell and on the other end, but really the critical one is the one that connects them, which maybe only has 2 connections. And if you can sever that dumbbell, maybe you can stop a pathological activity from spreading. Yeah, I completely agree that thinking about it in those ways can be really effective.
Hello, can you hear me?
Yes.
Yeah, this is Herman Cavalier from the NIMH. I'm curious, have they used DBS in epilepsy?
So I'm not an expert on that, but my sense is yes, that it has been used in some cases. Deep brain stimulation for epilepsy. I'm not sure of the parameters, but I think it has been used. There are also cortical stimulators that have been used in some cases for epilepsy, or I think that are being developed by NeuroPace, if not currently in use. But maybe someone else can comment if they know about DBS in epilepsy. Is Bill here? No, I don't see him.
Well, Bill sent me an email, Mark, and he wanted me to ask you. I guess he's listening on the webcast, so he can't talk directly.
Okay.
So his question is there any speculation about future connections to the lower levels of cellular activity and intervention at the neuropharmacological level?
Yeah, so that's ideally, I think, that's one thing we would like to be able to do, is to take something like invasive brain voltage data and somehow connect that to underlying mechanisms. And the path one way we're trying to do that is through the computational models. So you have a complicated data set, we have expert knowledge from the community, our computational models, and then trying to link the data to mechanisms through those computational models. So ideally, yes, we will be able to do that, but it'll take, I think, further methods development, figuring out how to make those connections, how to rigorously link a computational model and data to get to underlying biological mechanisms, and then suggest how to perturb those mechanisms to treat the disease.
So the whole electroceutical idea that's percolating, right?
What is that, electroceutical?
Yeah, if people want to Google that, I just read a Nature paper on it, and GlaxoSmithKline is actually holding some workshops on it right now.
Huh.
Electroceuticals. I can post it on the wiki as well.
Great. Yeah, so I think there is an important role for modeling, especially in this age of big data, of trying to take especially the knowledge of this community, the multi-scale modeling. How do we connect that to these large data sets that are going to come in, and then connect that data to mechanisms through our models?
Are there any animal models
Of what?
Epilepsy.
Yeah, so there are a bunch of proposed models, and it depends on the type of epilepsy you're interested in. So there are models of absence epilepsy. There are, I guess, maybe more dynamical or qualitative models, thinking about these attractor states. There are notions of changing an imbalance of excitation and inhibition and how that can cause epilepsy, or maybe gap junctions and how that can give rise to high frequency oscillations and then an explosion of seizure. So they range from the dynamical abstract models to the very detailed multi-compartment biophysical models. And I think it depends on the scale of data that you're interested in and the questions that you're interested in pursuing. And we're more at the dynamical, more, I guess, biophysically qualitative models.
Yeah, thank you. And I think I read there is also some pharmaceuticals. There is some that are used to avoid epilepsy, the crisis.
Sure. Yeah. So my sense is that there are many types of drugs that can be used to try to prevent seizures, and they can all act on different mechanisms, and it can be patient specific and seizure specific.
Oh.
Some drugs work for some patients, some don't. Some drugs don't work at all for patients, and then they may have resective surgery or brain stimulation instead. And some patients, they go on and off drugs that sometimes work and sometimes don't.